Thank you very much for joining us. We just wrapped up our session on COVID-19 and vascular complications as part of the Vascular Discovery 2021. I'm Pradeep Natarajan. I'm the Director of Preventive Cardiology at Mass General Hospital and Associate Professor of Medicine at Harvard Medical School.
And I'm Naomi Hamburg. I'm an Associate Professor at Boston University School of Medicine, where I direct the Cardiovascular Institute and the section of Vascular Biology, and I'm the immediate past chair of the AHA PVD Council.
We have three wonderful speakers for this session. They're going to give a recap of what they spoke during the session, and then we'll have a very brief discussion afterwards. So we're going to start with Dr. Anna Randi from Imperial College.
Thank you very much. So our focus is blood vessels and particular endothelium in blood vessels. And blood vessels became very early on in the pandemic, it became clear that COVID-19 causes severe vascular damage, which is linked to the thrombosis, that is a frequent serious complication, but also to the inflammation that is also causing a lot of the clinical features. And so we're interested to understand, there's a lot of controversy as to whether endothelial cells are targeted directly by the virus or not. And there's a lot of work in this field, and the jury is still out, but there's some really very interesting basic research at the single cell level, which will hopefully answer some of these questions soon. And from a translational point of view, the mechanisms that have been identified by several groups that show how endothelial cells are activated in severe COVID-19 patients are now presenting opportunities not only for biomarkers to stratify patient risk and predict outcome of therapies and so on, but also as therapeutic targets. And some of these pathways we discussed this morning. So it's a very interesting, it's a space which is very central to the disease and to possible therapeutic applications.
Great, thank you so much. Next, we have Dr. Yogendra Kanthi from the University of Michigan and NHLBI.
As Dr. Randi mentioned, we've been interested in understanding how inflammation might be driving some of this disease. And what we've come to learn over the last 18 months is that COVID tends to trigger hyperinflammation in some people. And while inflammation is good, it's important to try and protect yourself and fight off infection. If it's hyperactive, it can actually start to cause organ damage and tissue damage. And this really does seem to be the case in COVID as well. We in our group and a number of others have identified that there are blood cells circulating, immune cells that become hyperactive and start to cause clotting and cause more inflammation. There are platelets, which are the cells that circulate around and plug up holes in arteries and veins. But if they start doing that inappropriately, they start to cause clotting as well. And the same thing happens with endothelial cells, as Dr. Randi just mentioned. We've also found that there's an autoimmune response in the body. So COVID is triggering the body to form these rogue autoantibodies. These are antibodies that attack our own cells and tissues and cause destruction of those cells and tissues and can start to cause blood clots as well. So this is an area of a lot of interest for a number of groups now, and I think we're glad that some of this work and findings have led to clinical trials for patients. The real challenge here for us moving forward is going to be trying to understand when is the right time to try and suppress this inflammation, so that you hit this fine balance of being able to allow your body to fight the infection, but not suppressing it so much that you succumb to the infection. And then what happens after you recover from the infection? This immune system, now that it's got this hyperactivated state, does it have a memory to it, or does it all go back to normal someday?
Great, thank you very much. Last but not least, Dr. Clyde Yancy from Northwestern.
Thanks once again for allowing me to be a part of this. What a fascinating session. Thinking about the focus on the endothelial cell, how actively engaged. Is it a bystander and it is a consequence of off-target effects, or is it a target? And thinking about understanding the thrombotic cascade much more elegantly and identifying other precise targets that we can use so that we can change the consequences of the COVID-19 infection, or more correctly, the SARS-CoV-2 infection. That's where the clinical trial comes into place. This is exactly the conversation we had with everyone this morning. What's novel is not just doing the clinical trial, which usually is really hard work and it takes several years to execute and explore even a simple hypothesis. Now, because of a brand new way of executing clinical science, we've developed a novel platform. It is a connection of already existing networks, and we've seen some benefits that are quite striking. Yes, we've answered questions about the treatment of COVID-19, and we've fundamentally changed the care of patients within the first year of executing this. That's for the good. But more so and more emphatically, yes, we've demonstrated that there's a different way of doing clinical trials, of being more egalitarian, of being more inclusive. We've got diversity numbers we've never had before. We've tested over 85 agents for feasibility in less than a year's time. We've enrolled 6,000 patients in less than a year's time. If we can take those lessons learned going forward, then the new discoveries that Yogen and Naomi are working on, we can introduce those into these platforms quickly, answer questions precisely. But the other diseases that Dr. Natarajan and I think about every week can then benefit from a brand new platform of discovery. So I think this has been a really good news day for us.
Thank you all so much for this really impressive illustration of the power of science moving from basic models, translational studies with clinical samples to clinical trials, and really a model for transforming how we're going to do cardiovascular sciences in the future. I wanted you to all think about what is the next big scientific question that we're working on in the field of the vascular intersections with COVID-19.
Why don't we go in reverse order and let me do the clinical piece first? Because what we really need to understand, to be even more precise with our clinical trials, is whom is it that's at risk? We talk about phenotyping all the time. We talk about proteomics all the time. But can we use new research methodology to say, a priori, because of this profile, based either on genotypes or phenotypes, or maybe even some new machine learning logic, we can say these are the communities, these are the patients that are most likely to experience the complications, and then a targeted clinical trial, smaller numbers, shorter time span, because we know it's a high-risk group, and maybe even using surrogate endpoints, so we don't have to belabor the point for 1 or 2 years to get a question answered. So I think just starting up front saying what the clinical trialist needs is more refined targets, and I think that will be driven by science.
So I agree with Dr. Yancy. I'll echo those comments. As a vascular medicine physician and a cardiologist, one of my drivers in my research program is the diseases that perplex me in the patients that I see. And so trying to understand why some people clot and some people don't, or why some people develop vascular disease where it might be atherosclerosis and some don't is a burning question. And I think that the same can be applied to COVID as well. Why do some people have a worse response whereas others are asymptomatic and walking around? And then trying to understand how the vasculature interacts with the virus and the response to the virus is going to be important. And then understanding what drives the differences in response between individual to individual will be really helpful moving forward. So I'm looking forward to that from a clinical and a translational perspective.
So I guess as a final comment, all of these points are really important, understanding whom to treat and when to treatment. And one thing we have not mentioned, but I think that can play an important role in all of this, is the genetics of the response to the COVID-19, the SARS-CoV-2 infection and COVID-19 disease. And that which brings us back in a circle of understanding how it all works so that we can then also understand the predisposing factors to a different type of clinical outcome and understand how to better treat which patients.
I will maybe finish with a message, a public health message that Clyde Yancy brought up into the talk, that just the importance of vaccination and getting both all of you and all of the world vaccinated as our best tool that we have in our toolbox currently to combat this disease. And with that, I will thank all of you and thank the Vascular Discovery's organizers and the Heart Association for this wonderful session.